Hi there, welcome to AP Biofun with Dr. D. And today we're going to talk about accuracy of DNA replication and how the ends of chromosomes get replicated. In the previous lecture, we talked about the mechanism of DNA replication and how the structure of DNA poses specific problems for the replication machinery and how they're solved. Well, let's talk about accuracy and errors. So the fact that DNA polymerase works in the 5' to 3' direction and it needs a correctly paired nucleotide ensures that it is actually very, very accurate. So the type of errors DNA polymerase can make are really 1 in 10 to the 5th or 1 in 100,000 nucleotides. It is possible that the DNA polymerase can make. However, to this, we need to also add the fact that DNA polymerase can actually backtrack in the 3' to 5' direction. It has an exonucleolytic proofreading activity, which means it can go back and remove mismatched nucleotides and then insert the correct ones. That gives you another level of accuracy, which is 1 error in 10 to the second. In addition, most cells contain something called a strand-directed mismatch repair, a whole DNA repair machinery which is dedicated to detecting mismatched nucleotides and repairing them. That is another 1 in 10 to the second error rate or accuracy. So overall, when you combine these 3 mechanisms, the accuracy of DNA replication is 1 error in 10 to the 9th nucleotides, 1 in a billion. Now, this is very, very accurate, and this is great that it's accurate, but you also need to remember that we contain millions and millions and millions of nucleotides. We have a large genome, and our cells undergo many divisions. From the moment we have a fertilized zygote to a fully developed grown human organism, many cell divisions occur. So over time, errors in DNA replication will result in mutations, despite the fact that the process is extremely accurate. So again, just to remind you that DNA polymerase, in addition to being able to synthesize a new nucleic acid in a 5' to 3' direction, has a proofreading exonuclease activity, 3' to 5', which means it can go backwards and it can correct itself. So whenever it detects a mismatched nucleotides, the DNA polymerase will stop and go backwards and chew back any mismatched nucleotides. So how is accuracy of replication achieved in summary? Well, the DNA structure, which is a double helix with complementary bases. So if you make a mistake on one strand, you still have the original strand as a blueprint, and you can use that to correct the mistake. The fact that DNA polymerase cannot start a chain from scratch. It needs a correctly base paired nucleotide with a free 3' hydroxyl end. And the fact that DNA polymerase has the ability to correct itself, really. So it can go backwards, it can self-correct by chewing back incorrectly base paired nucleotides and then replace them with correctly inserted nucleotides. Now there's one last problem that is posed by the structure of DNA and DNA replication, and that is the problem of finishing the lagging strand, the one that is discontinuous at the very end. Now remember that eukaryotes have linear chromosome. That means we have a beginning and an end to a DNA molecule. They're not circular like in bacteria. So when the last RNA primer is removed, there's a portion of the DNA that has not been duplicated. So replicating the ends of the DNA molecules is a problem. So what happens when the last primer is removed? Nothing. The strand cannot be finished. And literally each time DNA duplicates, the ends of chromosomes are going to be shorter and shorter and shorter. So the three people whose work helped answer the questions, how do the ends of chromosomes get replicated, are Elizabeth Blackburn, Kara Greider, and Jack Shostak, and they won the Nobel Prize in Physiology and Medicine in 2009 for this seminal work. So how do the ends of chromosomes get duplicated? Chromosomes have something called telomeres. Telomeres are the end of linear chromosomes, and they're covered in special proteins. So telomeres is actually the DNA and the proteins it is covered in. What I just told you a few minutes ago is actually true. Telomeres will shorten each time DNA is replicated. So when telomeres shorten, chromosomes are shorter. So what do we need to make sure? We need to make sure that telomeres do not contain any essential DNA, any essential information. So telomeres are special DNA sequences at the end of chromosomes. And they're usually, let's call them nonsense. They're not really junk DNA, but they're like more of like nonsense. They're simple sequences which are repeated 100 to 1,000 times. They do not contain any genetic information. Why? Because you don't want to be losing genetic information each time the cell divides, right? So they're simple repeated sequences that do not contain genetic information. So when they shortened, the cell doesn't really lose any essential information. Now the telomeres are covered with special proteins so that the cell actually recognizes them as the end of chromosomes rather than a break in the chromosome. And again, we lose telomeres with age. Each time a cell divides, the telomeres shorten and shorten and shorten until they reach a critical length. Once they reach a critical length, that would signal the cell to stop dividing. As telomeres shorten and shorten over time, eventually they'll shorten to a point where this will be a signal that the cell needs to stop dividing. And if you remember what we talked about in Unit 4, how cells at the G1 checkpoint, at the restriction point, have to decide what to do, whether to divide, whether to differentiate, whether to die, or whether to retire to senescence. Well, that is the signal that signals the cells to retire. The cell will no longer be dividing, it'll retire, it'll grow old. Why? Because the telomeres have shortened, and that is a signal that the cell needs to stop dividing. So the length of telomeres determines how many times a cell can divide. Each cell and cell type has a limited number of cell division in itself, like kind of pre-programmed in terms of length of telomeres. So most adult cells have a limited lifespan, so they can divide a limited number of times and then they'll just sit there and grow old and eventually die. However, some adult cells need to divide constantly, like the skin cells that produce hair, cells of the immune system, germ cells, and others. So how do telomeres keep up with fast division in adult cells? And how do telomeres keep up with lots of cell division in the embryo? Remember, we all start with one cell and then we're trillions and trillions of cells. How do telomeres keep up with these divisions? There is a special protein, or rather an enzyme called telomerase, and telomerase job is to maintain telomere length. So telomerase is an enzyme that can lengthen telomeres. Part of the enzyme is an RNA molecule, and that RNA molecule is complementary to the telomeric DNA. And I'm just going to remind you that the telomeric DNA is just simple nonsense repeats. It does not contain any genetic information. So telomerase contains an RNA component, which is complementary to the telomere. And the RNA can serve as a template for the synthesis of telomeric DNA. So telomerase performs essentially a reverse transcription. It uses the information of RNA to make a DNA molecule. And if you remember when I showed you the central dogma, that an exception to the flow of information from DNA to RNA and from RNA to protein is reverse transcription. So this is where reverse transcription is encountered. There is another place when we start talking about viruses, I will remind you of this again. But in every actively dividing cell of an organism, reverse transcription occurs through telomerase. So again, telomeres shorten every time the DNA is replicated. However, if you were to add telomerase, telomerase will enable telomeres to be lengthened. Each time DNA replicates, telomeres are being lengthened by the function, by the action of the enzyme telomerase that uses an RNA template. Now, telomerase is also very important in two essential processes, the processes of aging and cancer. Telomerase is normally active only when we're growing, when we're developing. Telomerase is very active in embryos and also in adult germ cells. Very few non-germ somatic cells contain telomerase. In a fully developed adult organism, telomerase is turned off, it is shut off, it is not active. So without it, cells and organisms have a limited lifespan. Telomerase, if we were to turn on telomerase, that can extend the lifespan and that can postpone aging because we would be able, our cells will be able to divide more times to renew. We would be able to renew our cells. However, the trick here is that telomerase is also abnormally active in immortal cancer cells. So there needs to be a balance between lifespan and aging and cancer. If you have cells that are able to divide indefinitely, they essentially become immortal and that can give rise to cancer. And again, I would like to remind you that errors in DNA replication can occur. DNA can also be damaged by external factors. So if we do not fix any errors in DNA or any damage that has happened, the next round of DNA replication will result in a mutation. One of the new daughter strands will contain a change in nucleotides as shown here in this model. So it will result in one of the cells carrying a mutation. So changes in the exact sequence of base in our DNA need to be corrected. Why is that? Well, remember we talked about how change is good because changes that accumulate gradually over millions of years are the basis of evolution. The diversity of living things, their ability to adapt to a changing condition, different habitats, it's really good for the species, it's good for the biosphere. However, in the short term, change is bad because it affects the short term of survival of an individual organism, and genetic stability is needed. Change can be detrimental. It can lead to disease. It can lead to lack of viability, sterility. Adaptability of the species is not a concern. So what are the consequences of genetic changes? And we're going to talk more about this when we talk about mutations, but this is sort of a preview. A single nucleotide change is called a point mutation and can lead to no protein being made because it generates a stop signal for protein synthesis. It can lead to an altered protein being made, a different amino acid is incorporated, which could totally alter the function of a protein or abolish it altogether. And in addition to a single nucleotide change, there are also more extensive changes in DNA sequence that can affect the structure of chromosomes and have more dire consequences. And the best example of a single nucleotide change is, of course, something you're already familiar with, sickle cell anemia. and the changes in the mutant adult hemoglobin. A single nucleotide, an A, is being changed to a T, leads to a different amino acid at position 6 and leads to sickle cell hemoglobin. Now, changes in DNA sequence or mutations can be heritable or they can be spontaneous. If the genetic changes and mutations occur in germ cells and sex cells, they will be passed on to the offspring, and every cell in the body of the offspring will have the mutation. When a genetic change and mutation occurs, however, in a non-sex cell, in a somatic cell, it will not be inherited by the next generation. It may have no immediate consequences even for the organism. However, with time and with age, some somatic cells will accumulate multiple mutations, not just one. And those mutations can give them a growth advantage over other cells. They can disrupt body functions, and in the extreme, divide fast, become immortal, and travel to distant parts of the body, also known as cancer, as we discussed in Unit 4. And cancer really is the result of accumulation of many genetic changes. And that's why it takes time to develop cancer. Cancer incidence increases with age. The older you are, the higher the chance that you will develop cancer. And all aggressive cancer cells have changes that lead to genetic instability. So all cancer cells have changes in them that themselves lead to more mutations happening. So mutations and changes will occur at higher frequency than normal. People which have defects in the DNA repair proteins have a higher chance of developing cancer at a younger age. So genetic stability is achieved through accuracy of DNA replication and repair of errors and damage. So how do changes in DNA occur? Errors during DNA replication that are not repaired. Exposure to certain chemicals, environmental or internal. Exposures to UV or ionizing radiation. And of course, defects in the DNA repair machinery, which are either inherited or related to age. And remember that in order to maintain homeostasis, it is somewhat of a trade-off. On one hand, we want stability. We want to be able to accurately transmit our genetic information to the next generation with little to no change. On the other hand, we also want genetic variability, the yin-yang, because the genetic variability will generate variety. No two offspring are the same and allows organisms to adapt to changes in their environment. And this is all for now. See you next time.